Hello, everybody. We'll get started in just a moment. We're going to let everybody get in, and then we'll get started with the webinar today.
All right, imagine we are ready to start.
Awesome. Hello, everybody. I hope you can't hear my dog who decided to start barking as soon as the webinar started. Welcome to another Tennessee Urban Forestry Council webinar. This is our July edition. We are so excited to have all of you here today. There's a great turnout. We've got about 150 people. I see people already commenting about the dog. That's funny. We've got roughly around 150 people today, so we've got a really large crowd. I'm Imagine. I'm one of your hosts. I'm joined by Katie Donaldson and Kevin Bolger. And we're really excited to offer this course to you today. Before we get started and I pass over to Katie to talk about CEUs, I do want to point one thing out for all of you. For those of you that are in Tennessee or within the local surrounding states, for anybody that's interested in becoming an ISA certified arborist or is working towards that, the Tennessee Urban Forestry Council is hosting an ISA prep course on August 27th through 29th in Nashville, Tennessee at the Ellington Agricultural Center. So if anybody is local and they are interested in attending that course, it's already online on our TUFC website. And we can also post a link in the chat as well for you. But I'm going to pass over to Katie to talk about CEUs. Then I'll give you a quick introduction to Dr. Hoeksema, and then we'll get started.
All right, great. Let me share my screen here. Okay, good morning, everyone. So if you are on here to receive ISA CEUs or SAF CFEs, please make sure that you are registered and that you've paid the $10 fee. Also, just to help us out certifying who is receiving those credits, please make sure that your full name is showing, and you can do that by going to the Participants tab, finding the three little dots and clicking Rename, and please just use the name that you registered with. That just makes it easier for us again when we send that report to ISA and SAF. If you are a TUFC member, you do not have to worry about paying the $10 fee to receive those credits, or if you're just a general attendee and you're not applying for those credits, you don't have to worry about the fee. And if you have any questions, please enter those in the chat during the presentation, and we will get to those at the very end during the Q&A. We will also put up a little slide to help with the CEU reporting at the end and put a link in the chat just as another extra step that helps us out. And I believe that is everything. Imagine, I'll turn it back over to you.
Thank you so much, Katie, as always. And thank you so much, Dr. Hoeksema, for joining us today. The title of his presentation today is going to be Are Common Mycorrhizal Networks Beneficial for Trees and Forests? And Separating Evidence from Fantasy in the Wood Wide Web. So that's what we're going to be talking about today. Just a quick introduction on Dr. Hoeksema. He is a professor of biology at the University of Mississippi, where he teaches courses in mycology, ornithology, meta-analysis, and biometry. Very interesting subjects indeed, and we were talking about that before we got online with everybody else here. His research focuses on the ecological and evolutionary consequences of interactions among species, especially trees, and their mycorrhizal fungi. He has published more than 60 scientific journal articles, and in 2024, he received the University of Mississippi's Faculty Achievement Award for Outstanding Teaching and Scholarship. So I'll go ahead and I will let Dr. Hoeksema take it away. Thank you so much for joining us today, and please go ahead.
All right. Thank you, Imagine, and to the Urban Forestry Council for inviting me. I really appreciate the opportunity to talk to everyone today. I will go ahead and share my screen to get started. So indeed, I will be talking today about mycorrhizal networks, separating evidence and fantasy in the what's sometimes called the Wood Wide Web. I want to first acknowledge some co-authors and collaborators who have been important in the work I'm about to talk about. Justine Karst and Melanie Jones are professors in Canada at the University of Alberta and University of British Columbia, respectively. Francois Test is with a government agency in Canada, and Ian Mounts and Savannah Drought are both PhD students of mine here at the University of Mississippi. Also, I want to mention that this work was not funded by outside funding sources. All of everything I'm going to talk about today was just self-funded. And I also wanted to mention that for anyone whose Spanish might be stronger than their English, I have tried to include Spanish blurbs on most of the slides to help with communication. Okay, so first of all, I want to talk a little bit about some language, some definitions, and answer right away the question of what are common mycorrhizal networks. And I've started out here with an illustration, which is a cartoon where in green we have two different plants. In brown are the roots of those two plants, and in pink are mycorrhizal fungi connecting the roots of those two different plants. And that's what we mean by common mycorrhizal network. It's common between the two plants. To illustrate this a little better, I want to illustrate the contrast between simply talking about mycorrhiza, which is on the left, and common mycorrhizal networks on the right. So on the left, we have a tree, which is showing its relationship between its roots and the mycelium of a mycorrhizal fungus. Also, you may be aware, but many of the mushrooms that are in the forest are the fruiting bodies or reproductive structures of mycorrhizal fungi. But the body of the fungus is really below ground. It's the mycelium growing through the soil or other substrates, including dead wood, and absorbing nutrients out of those substrates. And it passes some of those along to the plant, to attached plants. These mycelia of the fungi are growing in an attachment to the roots of trees and other plants. And in fact, they can't grow without that attachment because they are fed by carbohydrates, which are the products of photosynthesis by the tree. And mycorrhizal fungi are completely dependent on these plants for getting their carbohydrates. They can't get carbon any other way, so they can't build their bodies without them. And so this is a mutually dependent relationship that is well established to be beneficial for plants. There are instances where the fungi are less beneficial or even detrimental, but in the large majority of cases, these fungi are well established to be positive things in a forest. But that's not what is the controversial scientific material that we're going to be focusing on today. Rather, we're going to talk about the phenomenon of a common mycorrhizal network, which is the connection between two different trees in a forest through the mycelium of these mycorrhizal fungi. So you can see that illustrated in blue here, connecting these two different trees by the mycelium. That's what we mean when we say common mycorrhizal network, or CMN. That's the phenomenon of focus today, focusing especially on that phenomenon in forests, connections between trees in forests through these CMNs. With that background, I want to set the stage for this project that I'm telling you about today. And that is that we are addressing popular claims about CMNs. This project was spurred by the overwhelming number of claims that we started hearing in popular media about CMNs, ranging from best-selling fictional books like Richard Powers' The Overstory, putatively nonfiction books, as well as very popular TED Talks, documentaries, magazine articles, newspaper articles, and even television series like one of my favorites, which is Ted Lasso, pictured on the bottom left, where Coach Beard is reading a book about fungi. And in these stories, we looked for and started to notice a series of especially three popular claims being made in these pieces. And in fact, I'll just go back and mention specifically what kind of put me over the top to really synthesize all of this was watching Ted Lasso in this episode. I kind of couldn't believe it that this topic had invaded my favorite television show, and while I really like that book that Coach Beard is reading there, that's the Entangled Life by Merlin Sheldrake. Coach Beard kind of misquoted the book and in fact made a statement that was really representative of the statements being made in all these other sources in the media that I really was sure at the time was false. And around that time I was also contacted by the author of this article in the New York Times to ask for a quote about this material. And I realized that in order to be really sure in my responses and my commentary on this topic, I needed to do a deep dive. And at the same time, Justine and Melanie were starting on the same kind of path from a different perspective for their own reasons, and we decided to collaborate on this and work together. What we identified ultimately were three popular claims that we wanted to address. First of all, that CMNs are really widespread in forests. Secondly, that CMNs are beneficial for trees in forests. And third, that mature trees preferentially send resources and signals warning of insect damage to their young, their offspring, or their kin through CMNs. We ultimately wrote a synthetic review paper on this topic, which was published in 2023 in Nature, Ecology, and Evolution. And a lot of what I'm going to talk about is summarized from that paper. So let's go through these one by one. And what I'm going to do is sort of summarize the claim, but then talk to you about what is the actual scientific evidence. What do scientific experiments and observational studies that are published and peer-reviewed say to us about these claims? Are the claims supported by the scientific literature? So first of all, this claim that CMNs are widespread in forests. In fact, based on the biology of fungi and trees, we suspect that these CMNs frequently do form in forests. Fungi in the soil, these mycorrhizal fungi, are often genetically compatible with multiple individual trees at the same time whose root systems are not that far from each other, and they can form these connections. We've seen it in laboratory studies, and you can even see it in the field if you are careful. But the question is, how long do those connections between trees last? How persistent are they, and how functional are they? So the strongest approach to actually verifying the contiguous connections of one fungal species colonizing multiple trees in a forest is using what we refer to as genotype mapping. And this is where genetic techniques are used to identify not only what species are involved, but actually the genetic individuals involved, so that you can find if the same genetic individual fungus is found on the roots of adjacent trees. So you can find, say, two different tree individuals where they have the same fungus individual on their roots. And if that is true, the implication is that at one point that fungus must have grown from the roots of one tree to the roots of another, at least temporarily, forming a CMN. And one such study I'll highlight, this is from Kristen Byler. It's a really nice study in a 30 by 30 meter plot in Douglas fir forest in the Pacific Northwest. This is from 2010. And what they did was they utilized these genetic techniques and found that two different species of rhizopogon, which is a fungus that forms a truffle and also mycelium in the soil, They looked at rhizopogon on the roots of Douglas fir, and the map on the right, the lines on the map are potential connections between different trees through mycorrhizal fungi. It's connecting trees. These lines connect different trees that had the same genetic individual on their roots. And one fungal genotype spanned at least 21 meters. So they found the same genetic individual of this fungus on roots that were 21 meters apart on different trees. So that suggests that at some point there were long fungal networks between these trees. So this is really a cool study, very impressive type of work. But what I want to point out is that those results are compatible with this model, this cartoon that I showed you before, where the same genetic individual of a fungus is found on the roots of two different trees. But those results are also compatible with this model, where the same genetic individual of a fungus is found on the roots of two different trees, but no longer connecting those roots. And this highlights the major uncertainty that we have about this claim, and that is that we have really no idea how long these connections last. Do they form temporarily and then get broken? Breaking of these networks in the soil is very common due to animal movement through the soil, breaking the mycelia. Parts of the mycelia die periodically. So it could be that what we're seeing in these genetic studies is the remnants, the legacy of previous CMNs now leaving the same genetic individual fungus on the roots of multiple trees that are near each other. And we really just don't know which is more prominent, how long these connections last, and we really need temporal studies to look at the time course of the formation and breakdown of these networks before we understand how widespread these networks really are in forests. So both of these models are compatible with the results from the genetic mapping studies. So we are left with this question of, whether hyphal links between trees persist long enough to be functional. In addition, I want to mention that there have only been 5 of these genetic mapping studies published to date. And only 2 tree species have been included of about 73,000 tree species worldwide, and involving only 3 different ectomycorrhizal fungal species out of more than 10,000. So, that's just a limitation of the studies that have been done. It's not a critique of these studies, which are very difficult to do and very impressive and valuable. It's just that we have such limited information about what is really known so far. And finally, the total area that's been mapped by these genetic maps is about 1 hectare total, out of about 4 billion hectares of forest that exist in the world. So our knowledge is really limited. Okay, so in summary, claim 1, the support is really limited owing to the paucity of information on CMN structure in the field. What we do know is that adjacent roots are often colonized by the same species of mycorrhizal fungi, and therefore CMNs are expected to form in forests. But what we lack is the evidence that they are widespread around the globe and persist long enough to be functional. Okay, let's move on to claim number 2, which is that CMNs are beneficial for trees in forests. So a common type of experiment that has been done to test this claim is utilizing seedlings, baby trees growing in forests that have been planted into experimental units, especially experimental units that differ in the presence of CMNs connecting the baby tree to an older tree, or potentially connecting them. That's indicated by plus CMN here. And they differ, these treatments differ in the presence of overstory tree roots, and whether those tree roots are able to penetrate and overlap with the roots of the tree seedling. So you typically have 3 different treatments, plus CMNs and plus roots. This is essentially the normal state of a seedling in a forest. This is natural. This is what a tree seedling experiences. It's connected potentially to the CMN of a tree, and its roots overlap with the roots of the overstory tree. In the middle, we have a tree seedling growing in a mesh bag or compartment that has a mesh that is something like approximately made of a mesh that has holes in it that are about 35 or 40 microns in diameter. And those holes are large enough to allow the purple to come through. You can see that here. That's the mycelium of the CMN. But those holes are small enough to exclude roots, which are bigger. So the roots of the overstory tree are kept out. So that's why this is a plus CMN minus roots treatment. And then the third treatment here is minus CMN minus roots, because the tree seedling is growing in a bag or a cylinder or a trench that cuts both the roots and the CMNs off and doesn't allow them to form. So you have minus CMN minus roots. This could be a small mesh, like 0.5 microns, or it could be a physical cutting. And by comparing the two treatments on the right, by measuring the growth and survival of the seedling in both treatments, you can test the effect of the CMN in the absence of roots. So if the seedling grows better, for example, in the middle treatment than in the right treatment, that suggests there's a positive effect of CMNs. As well, you can compare the middle tree to the tree on the left, compare the growth or survival of those seedlings, and look at the effect of roots. If the tree in the middle performs better than the trees on the treatment in the left, that suggests that roots have a negative effect on the growth of seedlings. So if you add roots and the tree grows worse, that means that roots are competitive. They're having a negative effect on seedling performance. So this is a really fairly common type of experiment that has been done. One problem, though, is that these treatments can have confounding effects that can really cloud their interpretation. So the treatment on the right, for example, this barrier here that cuts off the network may also reduce the total space in the soil that is available to the mycelium or hyphae of the mycorrhizal fungi of the seedling. So look at the space that that seedling has for its mycorrhizal fungi to forage, and compare that to the space in the soil that the middle seedling has for its mycorrhizal fungi to forage for nutrients. It's much larger, regardless of whether there are connections to overstory CMNs or not. And that could result in better growth in the seedling in the middle and an apparent benefit of CMNs, when in fact it may just be beneficial to have more soil to forage in. So that's one problem with these experiments that is really hard to get around unless you use a very large volume for this cutting treatment on the right, which is usually not done. So let me show you, let me show you a second confounding effect that we think is probably fairly common as well. The other thing that can happen is that these treatments, the mesh and the cutting, can change the community of fungi that are there. It can change which species of fungi are colonizing the roots. Some mycorrhizal fungi readily colonize seedlings in this situation where they are cut off from other mycelium. These are often fungi that grow from spores. And then in the middle treatment, you might get fungi that readily colonize seedlings by growing from mycelium connected to trees. That could cause these seedlings to be colonized by different species of fungi that have different effects on growth. So instead of you seeing differences in growth due to CMNs per se, it could just be differences in growth due to the seedlings being colonized by different fungi that have different growth promotion of their hosts. And in fact, out of all the experiments that have been done, 4 studies have measured this and looked for changes in the ectomycorrhizal fungal community as a result of these mesh treatments, and 3 out of the 4 studies found that the treatments changed the fungal community. And what about non-mycorrhizal fungi? There are pathogens, there are decomposer fungi in here, and we actually don't have any studies that have measured the effects of these treatments on these other fungi that could also affect nutrient dynamics in the soil and the growth and performance of tree seedlings. Let me show you one example of an experiment that has been done like this. This is from Ecology, from the journal Ecology in 2009. This is a really nice experiment, and I should note the lead author here is Francois Test, who is one of my collaborators. Melanie Jones is also a co-author on this paper. And in general, I want to use that opportunity to highlight the fact that we did not shy away from re-examining our own studies. I've done experiments like this, and so have all of my co-authors, and we included our own experiments in this reevaluation of the literature. And we agreed to take a hard look at our own work as well. So we're not calling out the work of other people exclusively here. What we're doing is examining the evidence that all of these studies present, including our own. This is a great experiment, by the way. I really respect it. It's perhaps the most thorough experiment that has been done on this topic in terms of testing mechanisms. If you look at the results, though, what they found is no effect of access to CMNs on Douglas fir seedling survival, growth, physiology, or carbon and nitrogen transfer from adult trees. The plus CMN treatments are found in the middle here, and statistically, they were not different from the various minus CMN treatments. In fact, the biggest effect was the presence of roots, which were potentially having a positive effect. Where you can see the highest performance, this is survival of seedlings, was in the no mesh treatment on the far right, where roots were allowed to access the seedlings. So this is where understory seedlings were overlapping with the roots of adult trees. When roots were cut off and prevented from growing into the cylinders and CMNs were cut off, we saw the lowest performance, but it's not statistically different from these middle treatments here. And by the way, remember that this 0.5 micron mesh may have been restricting the volume of soil that could be explored by those seedlings. So is this performance here lower because this seedling doesn't have access to CMNs, or is it because it has a lower volume of soil to explore overall? And this is one of those experiments where they found, in fact, that the mesh treatments altered the ectomycorrhizal fungal diversity found in the treatments. And indeed, the authors themselves concluded that they were uncertain whether resource transfer is an important mechanism underlying seedling establishment in response to these results. So that's just one example. That example is pretty representative of the body of work that's been done. We found no studies like this where all of these confounding factors had been ruled out, allowing us to conclude that CMNs had beneficial effects on seedling survival or growth. But let's say that you ignore those confounding effects, ignore those potential confusing treatment effects that these mesh bags and cylinders can have, and just assume that the experiments tell us what we want them to tell us, which is the effects of CMNs and roots. What do we find if we look at all the results together? So my PhD students, Ian Mounts and Savannah Dowd, have been conducting a meta-analysis where they pulled together all the published experiments from field studies on trees in forests to look at what do the experimental results tell us across all these studies. They found 13 scientific papers, including 41 different experiments. Altogether, these papers provided 243 different estimates of CMN effects on seedling biomass and 145 estimates of root effects on seedling biomass. And that's all. I'm just going to show you the results for seedling biomass, the growth of seedlings here. And these were mostly on ectomycorrhizal systems, not arbuscular, for those of you who know what that means. And I can talk more about that later if needed. In all of these studies, they calculated an effect size, which is the mass of seedlings in a plus CMN treatment relative to the average mass of seedlings in a minus CMN treatment. And that ratio should be larger when seedlings benefit from CMN access. And by taking the log of it, you kind of linearize it and make it easier to interpret. And they also are considering contextual factors that might affect these results, like which plant and fungal species were involved, and which type of mycorrhizal was it, the distance from adult trees, etc. I just want to show you some of the preliminary results. And on the x-axis here is the CMN effect size. To the right of 0 is a positive effect of CMNs on tree growth. And to the left of 0 is a negative effect. So all of these dots to the left of the blue bar are negative. To the right is positive. And at the blue bar and clustered around it are all neutral effects, basically no effect of CMNs on seedling growth. So the takeaway here, as you can see, is that it's a shotgun across a whole spectrum from positive to negative effects, and a lot of effects near 0. So we can't conclude from this, of course, that CMNs generally help trees in forests. Even ignoring the problems with these experiments, the data show us that sometimes CMNs are negative and actually hurt the growth of seedlings, and often they have barely any effect at all. We also pulled together data on the root effects. This is comparing seedling performance with and without the presence of overstory tree roots. And you can see that these results tend to be in the negative, but again, it's really a smear of data points across positive to negative. Sometimes roots are beneficial for the baby trees, and sometimes overstory tree roots are negative and hurt the growth of these young trees. And often they are neutral and around 0. So to me, what this means is that the question isn't yes or no, black and white, are CMNs beneficial or not, and are roots beneficial or not? It's clear here that the effects, if they are real, the effects vary a lot from positive to negative. And what we should be asking is, when are they beneficial? When are they negative? And when are they not having any effect at all? Under what conditions? That's going to be the really interesting work going forward in this area. Also, we wanted to look at what is the relationship between the CMN effect and the root effect in a particular study. So each data point here is giving you both of those for a particular study. And what we have found is that the relationship seems to be generally negative. So when you find more positive CMN effects, you tend to find more negative effects of roots. What this implies, and vice versa, and what this implies is that these tend to offset each other. So even when you find situations where CMN effects tend to be positive, the root effects, which are happening simultaneously, tend to be negative and offset them. And in reality, remember, tree seedlings in forests are not just enjoying the beneficial effects of CMNs if they're there. They are also, in reality, subject to whatever the effects are of overstory tree roots, which are often negative, and even when they're positive, they're offset by negative CMN effects and vice versa. So the net effect here is often neutral. So on claim number 2, in light of the evidence, first of all, alternative explanations are possible in all experiments that have been done to date showing putative positive effects of CMNs. And putative effects of CMNs are highly variable and often negative. And finally, putative root effects are negatively correlated to putative CMN effects, often offsetting each other. Okay, finally, I want to summarize the evidence for this third claim, which is that mature trees preferentially send resources and signals warning of insect damage to their young through CMNs. This figure from National Geographic from 2018 exemplifies the kind of message that's been in the popular media about this topic. If you read the fine print, you can see, that it's stating that the fungal threads connecting the trees are making connections that signal insect damage from, over on the right especially, warning signs. So through the network, trees under stress can transfer resources and chemical signals that trigger defense mechanisms in other trees. This is showing it between two mature trees in a wild forest. So what is the evidence for this phenomenon? In fact, we found no peer-reviewed published studies even directly testing these ideas in a field experiment. And there are unpublished theses from the University of British Columbia, for example, as well as greenhouse experiments that lack support for these ideas or even contradict this claim. So let me show you a couple of examples. This is an unpublished master's thesis from Suzanne Simard's research lab at the University of British Columbia from Amanda Assay, who was a master's student there, testing, and this was a really cool, innovative experiment, testing Douglas fir seedlings and their survival next to adult trees, adult Douglas fir trees. And the treatments that were set up here were using, first of all, kin and non-kin seedlings. So tree seedlings that were grown from seeds that were collected from that mother tree or that parent tree versus collected from a different tree. So either planted under the tree the seeds came from or not, kin versus non-kin. They also used mesh bag treatments to manipulate network access, and it was done at 3 different sites. Those first 2 treatments were crossed with each other to look at all 4 combinations. So a really nice experiment replicated across 3 different sites. I want to highlight a couple of results here. First of all, this is the overall effect of kin. And what you can see is that the dark gray bar here had higher survival, not quite statistically significant, but higher survival than the kin seedlings. So overall here, non-kin survived better under their unrelated trees compared to kin under their parent trees. Secondly, the effect of networks in the field here was non-existent. There's no difference in survival between seedlings that had access to networks and those that didn't. And over on the far right, we can see the interaction effect between networks and kin, which was also not nearly significant. If you zero in on that, you can see that if anything, this lowest bar, the lowest survival here, was in tree seedlings that were kin connected to adult trees through networks. The biggest effect here was a site difference. There was just a lot higher survival at one site than at the other 2 sites. So, if we sort of listen to the hype and focus on the potential effects of CMNs, we miss the main story here, which is that tree seedling survival is really affected by site differences, really basic forestry here that is having the biggest effect on the outcome. And the study itself, in the text, Amanda Assay concluded that our hypothesis that kin recognition is facilitated by mycorrhizal networks was not supported. But despite that, this study has often been cited in popular books as being supportive of the mother tree hypothesis and this idea that trees can warn their kin of insect damage and benefit them from it. The other study that is most cited as supportive of this phenomenon is a pot experiment from Song et al. In 2015 from the journal Scientific Reports. They grew Douglas fir seedlings and ponderosa pine seedlings uh, in pots where they were either separated or allowed to interact through CMNs um, under different treatment conditions. Under one treatment condition, they did not damage these trees at all. Under another, they allowed insect damage to uh, the Douglas firs. And under another treatment, they clipped the Douglas fir leaves to simulate insect damage. And what they measured in response was enzyme activity in the ponderosa pine tissue. Enzyme activities that may be indicative of defense upregulation of defenses. So if Douglas fir is warning ponderosa pine, then what you would expect is to see that when you damage Douglas fir, you see an upregulation of enzymes in the ponderosa pine. And that's what's on the y-axis and in the data over here on the right. So in some ways, these data fit the hypothesis. So if you look at the treatment of no defoliation, you see low enzyme activities in all three treatments, including the plus CMN plus roots, plus CMN minus roots, and minus CMN minus roots treatment. Those same three treatments we've been talking about. Low enzyme activity. And then intriguingly, when you damage the Douglas fir with spruce budworms, you see an upregulation of enzyme activity, especially in the plus CMN minus roots treatment in the middle. Not as much in the minus CMN minus roots treatment. And importantly, though, what I want to point out is that you do not see an upregulation of enzymes in the plus CMN plus roots treatment. Remember, that's the one that we call reality, because that's what seedlings experience in a real forest. CMNs and roots at the same time. And for some intriguing reason here, we see an upregulation of defense chemicals only when it's CMNs alone, but not when we allow roots to also grow over between the Douglas fir and the ponderosa pine. So I think these results are really fascinating, but it doesn't support the idea that CMNs in a real forest would allow warning of neighbors of insect damage, because the effect seems to go away when roots are overlapping. This is also true when the clipping treatment was used on Douglas fir. Again, you see upregulation of enzymes in the plus CMN treatment in the middle without roots, but when you add roots, which is the E bar down here, the effect goes away again. So, and that's it. Between these two studies, this is the evidence for this phenomenon that has been tested in the literature. So in general, we find no peer-reviewed published studies, especially in the field, supporting this idea. And the lab study here is intriguing, but doesn't support the idea that this phenomenon happens in a real forest. So whether signaling occurs through a CMN among trees in a forest remains to be demonstrated. I also want to quickly mention that our paper on this topic is not the only synthesis to look at these phenomena in the last couple of years. After our paper was published, these two came out with different groups of scientists also examining the issue from different perspectives. And then here is an editorial kind of pulling it all together from March of 2024 in the journal Nature, if you want to follow up and take a look at that. So I want to finish by talking a little bit about how to move forward on this topic. First of all, we've suggested that what we really need is a new generation of experiments. We don't think that this isn't a fruitful area for research, but rather we need experiments that, first of all, try to address the context dependency in apparent effects of CMNs. Try to understand that variation in effects from positive to negative to make sure that we understand when these might be beneficial, when they're not. Secondly, we need to keep in mind that it's important to understand that the relative importance of CMNs relative to other things that might be important, like roots, site effects, climate change factors like drought, and so on. And finally, we need experiments, of course, that control those confounding factors. And we do think those experiments are possible to do, and we provided some guidance on how those experiments can be done in our paper in 2023. And some of those experiments are being done now. And it's not a huge lift to do the experiments in a way that allows us to come to stronger conclusions about this stuff. Kind of a tongue-in-cheek conclusion here is that we need less hype, more hyphy. Focus on the biology, focus on what the data really tell us, and try to understand what is the overall true story that's coming out of the science of these things, rather than focusing on trying to tell a particular hyped up story that isn't supported by the data. And maybe most importantly to me, I think it's important to continue to tell amazing, compelling, true stories of forests and fungi. And there are plenty of these, and the evidence is strong for other stories about the roles of fungi in forests. And we need communication about forest biology that is effective and inspiring, but also grounded in the science, not sweeping contradictory evidence under the rug, but rather taking the whole of the evidence and telling compelling stories that get people and continue to get people excited about forest biology. And I'll give you just a couple of examples. One of them is the example of mycoheterotrophic plants, some of which are orchids, others of which are non-orchids. There are quite a lot of these plant species in the world. These are non-photosynthetic plants. Mycoheterotrophic means that these plants are getting all of their carbon from parasitizing other plants. And the way that they do this is through CMNs. So this is an example where CMNs do function absolutely to allow carbon to be transferred from one plant to another. These orchids are getting their carbon entirely through CMNs from overstory trees. And the evidence for this is really good. It's really exciting and cool because it involves orchids, which are charismatic plants. This is a well-supported phenomenon that we can talk about all we want, and the evidence is really good. Secondly, I want to highlight the story of truffles in forests. You know, truffles are interesting for all kinds of reasons. First of all, for their culinary value. They have a huge economic value. They're used in food. But truffles are the fruiting bodies, the mushrooms of mycorrhizal fungi in forests. And in fact, there are hundreds and hundreds of species of these truffles that are very common in forests. They make up some of the most important food for small mammals in forests. And this results in at least a three-part symbiosis or mutualism where chipmunks and other small mammals have a mutualism with the truffles where they're dispersing the spores of the truffles through their poop by eating the truffles and then the spores surviving through the gut of the mammal. And meanwhile, these truffles are having an important symbiosis with these trees. Remember, from the beginning of the talk, I mentioned that it's well established and not controversial that mycorrhizal fungi are beneficial for trees and forests. That's a fascinating relationship that most people don't know about. We can highlight that, talk about the phenomenon, and connect it with truffles and cute small mammals and orchids. And we've got plenty of material here to keep people captivated about forests and fungal biology in forests, well supported by the scientific evidence. So I'll stop there, and if we have time, I'd be happy to take any questions that you might have. Thanks for your attention.
This was incredibly illuminating. Thank you so much, Dr. Hoeksema. That talk was, I think, I'm not the only one speaking on this note. It was something that I think goes not against, but it brings a lot of light to what general thought is right now about what we believe of mycorrhizal fungi and CMNs, especially when you were talking about induced resistance, essentially in plants. So um, thank you very much. Hopefully we can have you back on in the future because I feel like you have plenty of other things that you're going to be sharing with us as more studies come out from your lab and your university. So I will leave it here to Katie. We have a lot of questions and we've got a lot of stuff to get through with 9 minutes left. So Katie, please take it away. I know you've got a lot to kind of filter through right now.
Yes, several questions in the chat. Thank you. And if anyone else has them, please enter those in the chat. I'm also going to share my screen for the CEU reporting if you just want to scan that QR code. And I think Kevin will also put a link in the chat if you need to get those CEUs. And that's just another way to help us. And well, hang on. I had the questions. There we go. Okay, first question. Is it true that many of the store-bought mycorrhizal inoculants can turn a potentially parasitic or detrimental under stressful conditions?
Mm, well, yes and no. So, store-bought mycorrhizal inoculants have been tested now in scientific studies and often shown to not work at all. Either the fungi that are included are dead or very limited in their mass so that they have no effect. Or more importantly, they are not collected from the local habitat or site where the plants are that are being inoculated with it. And what has been shown scientifically is that inoculants like this are generally only beneficial when they are developed from local material. Um, there's a group at the University of Kansas that has been testing these ideas in prairie restoration, for example, and finding that prairie restoration is more successful when locally sourced soils and fungal inocula are used to inoculate during the restoration process. So inoculants can be beneficial, especially if they are sourced from the local area or region in a similar habitat. But when they are not, you really need to be very, very skeptical of whether they're beneficial. Sometimes these products will show benefits only from the fertilizers that are included in the product. So yeah, look on those products with a lot of skepticism.
Next question. Do we know, or is there any way to determine whether the formation networks are purposeful or just hyphae associated with different trees interacting by chance?
Yeah, that's a good question. Well, what we think is that, it's important to remember that the fungi uh, have their own self-interest that they are behaving according to. Uh, Fungal strategies that are not beneficial to the fungi will have been selected against in evolution for, long ago. So these fungi, we think, are foraging through the soil to find plants that are giving them carbon. That's what the fungi need. So when the fungi grow through the soil and encounter a root, they don't necessarily discriminate against that root or purposely colonize it based on whether they're forming a connection from tree to tree or not. These fungi are connecting to trees that give them sugars, that give them carbohydrates, because that's what they need to get from the plants. Um, so yeah, just keep in mind the fungal perspective here, which is that the fungi need to do what is best for them, and that is to find tree roots and plant roots that will supply them with the carbon that they need to keep building their mycelium.
And before I get to the next question, I know several people are asking, saying the June webinar is listed when they click the link to verify for CEUs. Kevin, is it fine if they just go ahead and use that link still?
Um, yeah, I think it's just the title of the form maybe is wrong, but I'll double check to make sure that July option is there. But if it's not, go ahead and select June. We'll know that it's for this one.
Okay. Next question. What resources were accounted for in the resource transfers experiments, or what type? I believe this was related to the CMN effect and root size study. And someone also said their guess was sugars or carbohydrates.
Mm-hmm. Yeah, so there are a variety of studies that have measured potential resource transfer in the context of CMNs, where they've tried to manipulate the CMN connections and ask whether resource transfer happens more or less, whether the CMNs are connected, whether the roots are overlapping, or whether there's just a soil pathway. And those experiments, the best ones, are separating those different pathways where resources could move from plant to plant. They could move through the soil, they could move through roots, they could move through fungi. And the resource, to answer your question, the resources that are usually the subject of those studies include carbon, carbohydrates, where carbon, some sort of carbon label like a C13 or C14 is labeled in one plant, and then they look for it along the pathway and in the other plant. But also, there are plenty of studies that have looked at nitrogen and phosphorus, especially nitrogen, because it's fairly easy to use a nitrogen labeling to trace the movement of nitrogen from one plant to another. And sometimes water. You can put a dye, a colored dye, in water and look where it goes. And that's actually a really powerful technique, and that has been done as well. We have not seen strong evidence that CMNs facilitate resource transfer from plant to plant. For the same reasons I discussed earlier, all of the experiments have caveats where it's very difficult to test whether it's really the CMN or whether there are these confounding effects on resource transfer.
Next question, where the CMN effect is positive on the effect size is, where the CMN effect is positive, the effect size, I think they meant to ask, where it's positive, is the effect size more significant?
Hmm. Well, let me perhaps share a slide here just to help. Well, I'm not sure if I'm allowed to share. Okay. So I just want to quickly go back to the results of the meta-analysis and look at this here. So if you look at the results from the meta-analysis, this shows all of the different effect sizes. And I think what the question is about is these positive ones, are they significant? And the answer to that is that it depends on the study. And statistical significance depends not only on the effect size, how far to the right is the dot, but also the sample size. So a dot that is farther to the right is more likely to be statistically significant than one that's closer to zero. But it's also true that a dot that has 25 data points associated with it is much more likely to be significant than a dot that was built with 5 data points. So increased sample sizes increase the likelihood of statistical significance. So it's going to vary a lot among these individual data points, whether they were statistically significant or not. Some of the ones that are closer to zero here would be significant because they have more data associated with them. And some of the ones further to the right are going to not be significant because there were smaller sample sizes involved. So the answer just really varies among studies. That's the value of meta-analysis, because we can pull together all of these individual studies and we can come to conclusions based on the aggregate rather than having to worry about the sample sizes of individual studies. In fact, with meta-analysis, the way that we conduct this takes into account those sample sizes so that data points with larger sample sizes are more heavily weighted because they're considered to be a more precise estimate of the truth. So mathematically, the meta-analysis takes that all into account and helps you to come to broader conclusions.
And I know it's 1 o'clock now, so if people need to log off, we'll still count you as being here for your CEUs. Dr. Hoeksema, would you be able to keep answering questions?
Yeah, I'm happy to.
Okay, great. Um, Next question. Aren't these experiments for seedlings and therefore don't relate to CMNs in mature trees?
Yes, absolutely. I didn't mention that point, but it's an important point. All of the experiments that have been done testing CMN effects or putative CMN effects are measured, the responses are measured on seedlings, the growth and survival of seedlings. So even if you sort of ignore the confounding effects that we see in these experiments and accept the reality of the results, you do have to keep in mind that caveat that this is really most relevant for the performance of young trees, little seedlings. And we don't have any studies because for good reason, right? It's very difficult to measure the growth responses of adult trees over a long enough period to be meaningful. It's also difficult or impossible to measure the growth responses of large trees to do an experiment that manipulates the CMNs of adult trees. The closest thing that has been done is a correlative study looking at the diameter growth of seedlings, of adult trees in response to the size of the CMN that they're associated with from those genetic mapping studies. But only one of those studies has been done so far, and that's just a correlation.
Next question. In a weird way, does the balance net effect make some sense in terms of accounting for a balanced ecosystem in general?
Yeah, perhaps. I mean, just the fact that these trees exist with each other and the fungi exist with each other suggests that something is working out to allow them to coexist. Um, and and so the net effects here, if they were all super positive, that wouldn't really make sense, would it? If mathematically, if everybody was just helping each other all the time, populations would get out of control and then crash. So some negative effects are expected in real ecosystems, and um, we're certainly not arguing that it's all competition. It's clear that there are beneficial effects between trees and between trees and fungi. Those are important, but they're often offset by negative effects, and it's a complex web of positive and negative interactions that are ultimately coming together to result in the forests that we see.
Would it make sense that you would see more positive CMN effects with negative root effects as a compensation mechanism, or survival and vice versa?
Yeah, I don't really know. I can't think of a reason why they should try to offset each other or why that would happen. That's a really fascinating result from the meta-analysis that we have not been able to come up with a sensible mechanistic explanation for that. Why would you have negative root effects when CMN effects are positive and vice versa? I don't know. It could be, one explanation could be that when there is a transfer pathway from tree to tree through fungi or through roots, it's either one or the other. So you generally don't have both. And it could be that the hydraulics of soils and roots and fungi are such that they, the conditions tend to favor one or the other, so that it's one or the other, and maybe that is some explanation, but I really don't feel strongly about a good explanation for that phenomenon.
What about signaling threats by volatiles?
Yeah, good question. The evidence is much better in that area. It's certainly been shown quite clearly that trees and shrubs and other kinds of plants can emit volatile chemicals or do emit volatile chemicals, like jasmonic acid, for example, that can be detected by other plants. And other plants do detect those volatiles um, and sometimes upregulate their defenses against pests. So, yeah, and the evidence there is much better for those above ground volatile mechanisms. One example is, yeah, Laria tridentata, a shrub that's really common in the Great Basin in the western United States, from the lab of Richard Carbon at UC Davis, who was a professor there when I was a PhD student, has shown really clearly that when those shrubs are damaged by insects, they release jasmonic acid, and that other plants in the area can detect that jasmonic acid and upregulate their defenses in response. Now, it's important to recognize that, those trees or shrubs are not necessarily doing that, quote, on purpose to help each other. It's just what happens when they're damaged, certain chemicals are released. It may be that they've evolved a way to do this that ends up benefiting their kin, and that's a strategy that has worked. And if it's genetically based, then it would be selected for in over evolutionary time. But yes, the evidence is much better for those volatile kinds of mechanisms.
Is your lab studying these topics in other ways and other tests over time?
Yes. Um, for example, my PhD student Ian Mounts just completed an experiment in giant sequoia forest in California, testing whether adult giant sequoias may affect seedlings of giant sequoias through CMNs. And one innovation that he implemented was to add another mesh treatment that allows us to separate the soil pathway really clearly from the fungal pathway. And that will allow us hopefully to interpret those results with a little bit more clarity and fewer confounding effects. We also plan to carefully look at the fungal species in each treatment to understand whether the treatments may have um, affected the fungal communities in a way that alters the results. So, and also that experiment was done in an arbuscular mycorrhizal system, which is, most trees are, most of these experiments have been done with ectomycorrhizal fungi, so that is one way we're trying to move it forward by studying these things with arbuscular mycorrhizal fungi a little bit more. And that study will hopefully be published in the next couple of years. We also have an experiment that we've done in local oak forest here in northern Mississippi, where both ecto and arbuscular mycorrhizal fungi are potentially involved.
Okay, I know we're getting close. We're at the 110 mark. Just a couple more questions, if that's all right with you.
Sure.
Can VAM, arbuscular fungi, communicate? Is it possible?
Hmm. Well, so arbuscular mycorrhizal fungi have been studied regarding these phenomena. I do want to emphasize that my talk today and the evidence that we've gathered was focusing on trees and forests, and mostly in ectosystems, not arbuscular mycorrhizal systems, although there are plenty of trees that are colonized by VAM or AMF, arbuscular mycorrhizal fungi. So in temperate forests in our region, in the Mid-South here, for example, maple trees have AM fungi, sweet gums. Oaks have both. Sometimes they seem to have more arbuscular when they're younger and ecto when they're older. And then plenty of tropical trees are dominated by arbuscular mycorrhizal fungi. So we need to understand those phenomena better. But most of these experiments that have been done have been with ecto systems. So with trees anyway, we understand very little still about what might be going on with AMF, with arbuscular mycorrhizal fungi. So I would say stay tuned for the answers with trees. With herbaceous plants, there are some experiments that have been done that are really intriguing, but I'm not prepared to summarize those results today.
Will the fruiting bodies send these signals through their spores as well, a pheromone of sorts, or is there only evidence of these signals underground?
Good question. Before I answer that, just quickly on the AMF topic, I do want to highlight one experiment that has been done with tomatoes and arbuscular mycorrhizal fungi from Song et al. And that experiment is really pretty compelling, showing that damaged tomato plants may emit signals through AM hyphae that might allow upregulation of defenses in neighboring plants. But again, that experiment used the same methods we talked about today with the same caveats. And so we're not sure of the pathway there, whether it's through the fungi or through the soil. Okay, to answer the question, can the signaling potentially happen through mushrooms? Well, mushrooms, you should think of mushrooms as kind of fruit on the tree. The body of the fungus is really below ground. The mushrooms are sort of peripheral. They're attached to the mycelium, but they are above ground structures. It is really a cool thought, though, a cool question of whether there are volatile chemicals that are being emitted by mushrooms. And there's only very little research on that that's been happening. And I would love to know the answer to that question, but we just don't have the data.
All right, and this is the final question. As forest ecosystems are nonlinear in nature, are you aware of any research of quantum effects or quantum communication?
Hmm. I am not. Yeah, it's a good question. And I would say just generally that indeed forest systems are really complex, and there are probably phenomena in forests that are difficult to predict from the results of single experiments in pots, even experiments done in forests with tree seedlings over a short period of time. There are longer term phenomena, larger scale phenomena that are not predictable from those experiments. And we need to be keeping that in mind. On the other hand, all we have to examine is the evidence at hand. And we've taken into account all the evidence that we could in this synthesis so far. And if my colleagues who are more mathematically inclined can provide some data on nonlinear effects and quantum effects in forest, I would love to see those data and those results.
All right. Well, like I said, that was the final question. Thank you for taking the time to answer all those questions.
My pleasure. Thanks for the opportunity.
And I'm not sure if Imagine, she may have had to drop off the call. But thank you again so much.
Thank you, everyone. Have a good day, and we'll see you next one.